If everyone can come back in. I know one feedback we get every year is make the breaks longer for the networking, and they were a half hour this year, but they keep getting shorter as we keep discussing, and I feel like it's still never long enough. But let's come, if you can come back in, we'll start with our final session on how AI can help and real-world evidence can maybe help transform some of drug research and development and labeling options. So I'm going to pass the baton to Dr. Liang Li, who can present, introduce our speakers.
Hold on. So we have, first one have Dr. Zhao Liang from UCSF School of Pharmacy. So he's going to talk about AI, how AI can transform modeling approach in drug development. One thing I want to kind of highlight though is Dr. Zhao has been working in the FDA for many years before he joined the faculty over there. I think this is where we really want his experience on both governance agency and also in academia, and see how AI can help on the drug development. Go ahead.
Yes, thank you, Dr. Li, for the intro and the invite. It's my first time to be at Indiana University. Actually, I was a graduate from Ohio State, so really want to congratulate the school for the national championship. I'm a big fan. Growing up in Ohio State, you always live with the football. So as a Chinese, I'm a rare Chinese. I can understand every move of football. I can even predict, what will be the next offensive strategy based on the formation. Usually, my call is 80% correct. So as Liang just introduced, I worked with FDA for a total 15 years. Prior to that, I worked with industry like BMS, Medimune for biotech, and then acquired by AZ. Then I joined FDA initially as a clinical pharmacology reviewer, then became a team leader of pharmacometrics, covering oncology, rheumatology, pulmonary, inflammation. Then after that, Office Generative Drugs wanted to establish a division for quantitative method modeling. Then they called me in. So I served as the establishment division director for the last 10 years, collaborated with many people in the audience, including Dr. Robert Bies. Together, I think the most valuable investment, even though not much, is the investment on PyDarwin. Now it's becoming an industrial standard. Everyone is implementing that. So it's really, really a proud thing to collaborate with many top scientists in academia by then. So I still don't know if I have, okay, great. Actually today, my talk is less about regulatory. I think it's boring. That's why I left FDA. So it's regulatory is the most conservative place, right? Then I joined UCSF as a professor, based on, they recruit me. I don't know why they recruit me. They trust me. Someone from academia can do innovative work. They even gave me a professorship there. So after I got to the Bay Area, I was fascinated by the culture, or by the pace everyone's moving. Actually, yesterday, OpenAI released their a life science tool. It's called Roseland. I don't know if I've read the news yet. If you check the ecosystem and partners, you can, from one of the code, you can find mine. So I have been collaborating, the OpenAI just 200 meters away from my office, like Sam Altman. When you think about it, 200 meters away from you, you feel you are so close to AI. Then I collaborated with them. Actually, I was a consultant with OpenAI. They do want to have an entry to the farmer. And what would be the strategy? Then they dare to hire me. Then with that, in the Bay Area, I really see lots of emerging trend and kind of making the drug development really, really different from before. In the last 2-3 weeks, probably when you read the news, like Efficient Bio was acquired by someone with 400 million, and every week there's some news like a biotech AI company with even joint venture or acquired by someone, by a tech or by a farmer, or a farmer joint venture with a tech. I think that's going to be the future trend. So that makes me think, can I just synthesize what I have observed in the Bay Area and download that here? So the message is I think it's pretty new. So I haven't shared with anyone else yet. So I didn't do much rehearsal. So forgive me if I got stuck in the middle. So today, I think I'm gonna skip this slide because everyone knows drug R&D is costly. It take a long time, and once the drug enter to a clinical stage, only 10% of them will make to the patient. So with that, many people are thinking how to make the drug R&D pipeline more efficient. So actually, when you see in the Bay Area, there are lots of startups. So in addition, on top of the startup, you always hear AlphaGenome is out, AlphaFold 3, and Pro, RF diffusion. They are doing different things. So many in the drug discovery area. So I've been telling them that I have some reservations on trusting this model. They are maybe just working on 1 to 5% of the equation. If you have a small molecule that combines to the target perfectly, so that's only one small step in drug R&D. But however, I think they are really making revolutionary changes. So let's go with AlphaFold 3. I think you've been reading the news, right? It beat AlphaFold 2 in many aspects. I think that the main advance is that it can directly generate 3D atomic coordinate. So not only it can predict the folding of isolated protein, it can predict the protein complex, including can it co-fold with other proteins, small molecules, DNA, RNA, or even ions. So it gives you a more realistic picture of protein folding. So then SODD, from like a spin out of drug discovery unit from DeepMind, Google DeepMind, then they have, I'm sure you have read news about SODDE. SODDE is mainly like extend the capability of AlphaFold. It have structure prediction accuracy of native AlphaFold 3 on novel pockets. So mainly on the keywords on novel pockets. It can also predict the PK properties, based on a molecule or a binding. Can you predict the ADMET property of a molecule? So everyone has been trying that for a long time, right? Initially, the question is, when you see the chemical structure, can you tell whether it's a drug or not, whether it's have efficacy, safety issues? So I don't believe that story is true, being a drug developer. But they claim they can predict that. Then Pro is more like an enhanced version of SODDE to use like a physics-informed data to further advance the binding pocket prediction, to better predict the binding affinity of a small molecule to the target. So I think the data used to train Pro is much larger than the data used to train SODDE, because they use physics-based modeling simulation process to generate data for the whole chemical space. But for SODD trainings mainly used the known chemicals and proteins. So therefore, when you have a new target, you have a new pocket, and their data is scarce, then I think you need to look at the pro. Then David Baker, right, the Nobel Prize winner last year, then his contribution is that, so once you have a target, can you generate a protein that have never been in presence? So it's more like you are the creator of the world. So based on the structure or the pocket identified, and can you redesign, totally design de novo protein to bind to the target? I think it's another revolutionary concept. So I will skip this slide given the time. It's more like a foundational models summary. So what the models are doing. And I haven't fully explored the OpenAI's Roseland release yesterday yet. I don't know what the fancy things are behind that, even though they do give me access to their latest model. That's why my code is there. So we have meetings with them bi-weekly. Then I will download more, hopefully in the future when I get the chance. So now we're talking about AI agents in clinical development. That's where in FDA we have been reviewing most of the data from the clinical development. FDA actually doesn't care much about the preclinical data. It's mainly reviewing the benefit risk profile of a compound, mainly the phase 3 study. We call it registrational study, right? That's where every, the probability is based on, so it's sometimes called registrational study, sometimes it's called a pivotal study, the mainly phase 3 studies. Sometimes phase 2 could be registrational, pivotal, if the approval is based on phase 2 result, or phase 1 for some PD-1 drugs. By the way, I was the lead CLIFARM review team leader when both PD-1 products were approved, like Lumab and the others, pembrolizumab. So in the clinical development, what can AI contribute? It's about, there are so many companies in the Bay Area as well. Someone trying to get me into the advisory board. I can't decline most of them given the conflict of interest, because I want to do the same thing as well. So then you see lots of opportunity there. First of all, like a protocol design. So IQVIA, for example, NVIDIA, they formed a partnership. Basically, they do another type of trial simulation, basically based on the real world data and the historical data. Then IQVIA, right? They have a vast of data sitting in their house, and they simulate the trial. So with this inclusion criteria, can I exclude 80% of the patients? If so, modify the protocol. With this protocol, with this clinical endpoint, based on the real world data, is that too variable to drive any meaningful conclusion? If yes, modify the protocol. So they have really formed an efficient partnership. In terms of protocol development, they do trial simulation using real world data ahead of time before even write down the anything in the protocol. Then patient recruitment agent. When I was in FDA, like 10 years ago, if you attend a conference called the DIA, Drug Information Association, then you will see there are so many companies, they make a fortune by recruiting, by helping the farmers to recruiting the patients. The idea is that now we do not need to do that manually through network or connection. It's just you use AI to screen the EHR data and from the hospital, and also read the clinical notes, and use either NLP or lately more like large language model for UCSF. There's an in-house large language model called, we have been using that in more like a GPT 4.0 based. You just extract information from a clinical note to convert unstructured data to structured data. Then you also can learn the family history of patient. With that, you can recruit patients very quickly. Used to take a month to identify a patient. Now you can only take minutes to identify the patient to be recruited to the trial. That's another AI utility. Certainly, operations and report writing, you can use AI to do that. I believe all of the major pharmas, they're developing their internal capabilities to generate reports, everything, utilizing AI. It's the easy thing to do. Even in my lab, actually the former OpenAI biopharma lead, Aaron Jake, when he left the company, he actually worked with me for free. Then we did generate something even for population peak report. You can generate it immediately after the modeling. So I have been talking about AI for drug R&D, right? So, however, there's lack of success cases so far. People have been challenging, AI's been amazing for so many things. Why there's lack of impact? There's no impact cases from the drug field. Today, I just want to share two of those. Hopefully, that can stimulate some thought where AI can really help revolutionize drug R&D. The first case is about rontercept. So the drug is special and impact making is because the AI contributed to three things. First, the AI contributed to identify the target. Second, the AI contributed to develop, to generate the molecule against the target. The third contribution is that it shows the molecule does work. So it's for the idiopathic pulmonary fibrosis. That's the indication. And then based on the proteomics, transcriptomics, and all those data for this indication, they identified a new target that never been discovered before. It's called a TNIC. That's the target. And not only with they identify the target, then they use an internal AI platform to, like a generative AI platform, generated a chemical that can bind to the target. The chemical have never been existed in any chemical library. Then they put that into phase 1, phase 2 studies. Here you can see, based on the phase 2A result, the patient taking that test, the drug, have increased the PVC, like it's more like a measure for the lung function, forced vital capacity by 98.4 mil. So in comparison, in the control arm, so the patient declined, the PVC value declined by 20 mil. So you see a gain of about 120 millimeter. Is a significant achievement. In the past, all the medications usually only slow down the deterioration of the drug. So you can see it kind of a reverse. It's kind of make a patient recovering. It's amazing, right? So with that, I think probably we can see either this year or next year, probably we can hear some news about this drug. Another case is glasib. So this drug is also amazing drug that's been discovered by AI. It's utilizing it's for cancer treatment. It's utilizing PI3K alpha pathway. I don't know if you have been dialing into FDA advisory committee meetings when they have, in the past, this very popular target. But I think the pipeline for this target has been terminated within the farmers just because of toxicity, like the skin rash and hyperglycemia. I think this pathway has been built as by the farmer. We should abandon this pathway. However, the AI have identified amazing thing. In the past, when they are trying to screen a molecule to bind to the cancer cell with the target and the wild type target, they found they are statically the same. The structure based on the crystallography, they are the same. However, based on the AI simulation, so if you simulate the protein folding and the motion of the target, it's more like when you are practicing Tai Chi, even though the form is the same, but the disease and the cancer cells and the healthy cells are performing differently in one step. So there's a cryptid pocket temporarily in the motion of the target. Then they are different. Then they designed a molecule to specifically, when you see that gap, the cryptid pocket opens, then you insert the small molecule into it. Therefore, it only targets cancer cells. So with that, I think it's a relatively brand new concept in drug R&D. With that, they do, in the phase 1-2 trial, they do find a progression-free survival benefit, not a benefit. They do find the progression survival median time to PFS is 11 months. It's a really impressive finding. So this drug gained FDA breakthrough designation, meaning FDA is going to put some effort to zoom into the review of this drug and have a early, make sure it have an early entry to the market if it really works. So now we can see that based on the new development in the tech field and also the impact-making cases, we see a glimpse of light that AI is going to make something, in the future. However, what is limiting us compared to large language models that you can scrape loads of internet information to train the model? What limiting the AI use in drug R&D is the data wall. We don't have that many high-quality data. Now, they have a new solution, based on some physics-based modeling, which can really predict the drug binding and the structure well. They use those to generate the data, to enjoy the scaling law. So which company, whoever can generate those data, they're making a fortune right now. Another trend is that hybrid AI plus physics simulation. I think when AlphaFold 2 or 1 really shocked everyone. They are only using the empirical, like input-output type of thing. And now people are combining the physics law into the equation. They can increase the efficiency by many, many folds. Those are another trend in the field. So this is the last concept I want to share with everyone here, is that in the Eli Lilly, right? You heard Eli Lilly has collaborated with NVIDIA to form the most modern, architect for drug development. Meaning, given the lack of data, then the concept is that you use AI to design a drug molecule. Then you use robot, not a human being, to synthesize the molecule and do the assays. Then you use AI to conduct the data analysis. Then sometimes it's matched with your expectation, sometimes it doesn't. Then you use the data, experimentally obtain data to give that back to the AI to fine-tune the model. Again, it's more like it's called the autonomous closed loop. I think it's going to be the future R&D mode for the farmers. Okay, I think I'm I'm out of time. I will skip this slide. As I said, a regulatory landscape, and it's pretty conservative, but I see there are some guidances. Also, there's a first approved a tool, like a diagnostic tool to read the liver biopsy slice. That AI tool has been approved, meaning it's cloud-based, meaning every company can enjoy it. And with that AI tool, you do not need a human being to read the slide. Then you have a standardized output and an increase in consistency in terms of data generation. If anyone is doing something against the indication like a MASH, I think this probably the only achievement in FDA regarding the AI adoption. For other adoptions, that's being officialized, as a tool, a policy. Even though there's a guidance, I don't see much advancement yet. Certainly, FDA need to have a cumulative cases to make things happen. So this covers the flow of today's lecture. I don't want to repeat again. So I think the key takeaway is that 2026 marks the formal farewell to the manual trial and error era of drug development. I think we are entering the scientist in the loop era. As a researcher, in academia, so we need to pay attention to this. Certainly, that will involve like how to revolutionize the modeling approaches, how to validate, how to keep improving the modeling simulation tools. With that, I just want to show thanks to lab member Sangya Chen. I mentioned Aaron Jake had joined my lab for free to work together for developing some AI tools. Sangya is another meta engineer who worked there for 4 years and took very low salary, work with me. Hopefully we can generate something fun together. And also the people who trust me, I'm been in the FDA for so many years. People not sure if I can be successful in academia, but still people trust me, joining me in the effort. Now have lab size about 6 to 10 people. It fluctuates. So with that, I welcome any future collaborations and any communications if you would like to have with me. Thank you very much. Again, thank you for the invite.